The outline for my talk today is going to be to talk about what I think is a pressing need for diversity in the search for Alzheimer's disease treatments. I'd like to propose a new paradigm that we have initiated for a diverse and integrated approach for discovering novel therapeutic mechanisms in Alzheimer's disease. I'd then like to give you one example of how we've used this approach to understand the metabolic basis of pathology and progression in Alzheimer's disease, and then conclude very briefly with an overview of some translational implications of these findings. Just to put these studies in a historical context, it's been now more than 100 years since Dr. Alois Alzheimer described the pathological features and the clinical symptomatology that now constitutes what we recognize as Alzheimer's disease. Auguste Dieter was his patient, a 51-year-old lady that came to him when he was a practicing psychiatrist in Frankfurt. He took care of her for 4 years, and when she died, had the privilege of getting her brain after autopsy and doing very detailed neuropathological assessments using silver stains that are in use to this day, and describing the neurofibrillary tangle as well as the neurotic plaque, which are today recognized as the pathological hallmarks of this disease. An interesting side story here is that when Dr. Alzheimer first presented these findings, he presented them at a small meeting called the 37th Annual Meeting of Southwest German Psychiatrists in Tübingen, Germany. And the conference organizers, in their infinite wisdom, decided that these findings were really not interesting or novel enough, even to merit publications in the proceedings of the conference. Just goes on to show you that the system of peer review was as temperamental and quirky 100 years ago as it is today. But I always like to think about this story when I get nasty reviewer comments back, and I think I'm in very distinguished company when that happens. But despite studying this disease for more than 100 years, despite hundreds of papers having been published on the basic biology of the disease, what we have to offer our patients is precious little. So these are the 4 FDA-approved treatments for Alzheimer's disease. So these are merely symptomatic treatments. And some would argue that these are merely palliative interventions. The last of these was Namenda, that was approved by the FDA more than 10 years ago. What makes this situation a little bit more concerning is if you were to look at all of the drugs that are currently in preclinical development, and you can do this fairly easily, you can go to the ALZ Forum website, search for all of the disease-modifying treatments that are being developed, and stratify them by mechanism. What you see is a striking lack of diversity. You see that most of these medications, most of these interventions that are now being developed in the preclinical stages target some aspect of brain A-beta deposition. They either inhibit its deposition or accelerate its clearance or break up aggregates of toxic A-beta species. This is despite the fact that till today, there has not been a single clinical trial that has clearly established that this is in fact a valid treatment target. Far from it. So these are just 2 examples of 2 pivotal phase 3 clinical trials reported about a year apart in the New England Journal of Medicine, showing the failure of a gamma secretase inhibitor as well as a monoclonal antibody against A-beta in Alzheimer's disease. Some of these interventions have not just failed, but they've also worsened cognitive performance in patients with established disease. This becomes even more dire when you consider that each one of these molecules that have made their way into human testing in clinical trials, when you look at their track history of how they did in preclinical animal models, each one of these molecules seems to do a fantastic job of curing Alzheimer's disease in the mice. They clear plaques, they clear tangles, and where there's a cognitive or a behavioral phenotype, they also ameliorate symptoms. But every time you move these molecules forward into human testing, they result in resounding failures. So how do we bridge this yawning clinical translational gap between the promise of preclinical drug development and the grim reality of failed human clinical trials? This is one example of a review article that proposes that there is now a pressing need for us to rethink our strategy in developing disease-modifying treatments. And rather than focus on preclinical development in animal models, to try and leverage a much deeper comprehensive understanding of longitudinal changes in human physiology during aging, and use that knowledge then to try and identify plausible mechanisms for intervention. So how do we do this? How do we put this in action? So what we have proposed to do is exactly this, to try and leverage a deep, thorough understanding of longitudinal changes in human physiology during aging. And the way I think about this paradigm is how I'm going to present this over the next couple of minutes. I'm going to present this as a sequence of 6 questions, and we systematically answer each one of these 6 questions. The first question is you start with the risk factor of interest. And in our studies, we've studied several genetic as well as non-genetic and environmental risk factors. The environmental risk factors that we've studied include obesity, inflammation, insulin resistance, vitamin D deficiency, and most recently, exposure to anticholinergic medications. The emphasis in our studies is to study these risk factors as early as possible. So we've studied, for example, midlife obesity, midlife exposure to anticholinergic drugs. We've also been interested in studying a variety of genetic risk factors, and these are hits that large-scale genome-wide association studies have suggested are associated with risk for Alzheimer's disease. The second question that we've asked is, once you identify your risk factor of interest, we then want to test the associations of each of these risk factors with every phenotype that we've acquired during longitudinal studies of human aging. And these examples include risk for incident Alzheimer's disease, longitudinal changes in cognitive performance, brain atrophy, brain amyloid deposition, insulin resistance, and so on and so forth. We've done most of these studies in the Baltimore Longitudinal Study of Aging, and those of you who are not primarily in the aging field may not recognize this study. It's among the longest running studies of normal aging in the United States. It was started by Dr. Nathan Schock in 1958, and we've currently studied more than 3,000 participants. Our current enrollment is at more than 1,300 individuals. In 1994, Dr. Susan Resnick started a neuroimaging sub-study to the BLSA. And as part of the neuroimaging sub-study, we've assessed more than 150 participants annually using brain MRI and O15 water PET images. Since 2005, we've acquired longitudinal in vivo amyloid imaging in a subset of these participants. All of our participants give us blood samples that we can bank for biomarker studies. So a typical BLSA participant in the neuroimaging study would have undergone 10 annual brain MRI, 10 annual PET scans, and would have given us multiple serial blood samples during the course of their participation in the study. And what's remarkable is about half of our participants also enroll in an autopsy program, so that at death, we have access to their brains and can do very detailed neuropathological assessments. So this is really an extraordinarily rich longitudinal data set in which to ask some of these questions. So the third question that we've asked, after identifying the risk factor and testing its associations with AD-related phenotypes, is whether this risk factor changes gene expression both in the brain as well as in the periphery. And we think these parallel analyses in blood and brain are critical in determining the systemic triggers of AD-related neuropathology and symptom onset. Question number 4 is your risk factor of interest also associated with altered protein and metabolite levels in the brain? And here we've used essentially predominantly mass spectrometry-based proteomics and metabolomics. We've acquired both quantitative as well as global profiling of proteins as well as metabolites in the brain. We've used mass spectrometry-based methods to quantify well over 600 metabolites in the brain and label-free quantification of approximately 1,000 proteins. We've done these analyses in brain regions that are vulnerable to classical Alzheimer's disease pathology. So we've acquired data from the inferior temporal cortex, that's an early site of tau deposition and neurofibrillary pathology. We've also done these assays in the middle frontal gyrus, which is vulnerable to amyloid deposition, as well as in a brain region that's resistant to classical Alzheimer's pathology, so in the cerebellum. An important point to note here is that we've typically used in these studies 3 groups of individuals. Individuals with a clinical diagnosis of Alzheimer's disease that's confirmed at death, healthy control. So these are individuals who've had no cognitive impairment during life, and their brains, in fact, do not show any Alzheimer's pathology. And the third group of individuals that we've called asymptomatic Alzheimer's disease, which I'll talk about in just a minute. And that comes from the recognition that even though we understand that neuritic plaques and neurofibrillary tangles are the defining pathological features of Alzheimer's disease, there's now growing recognition that a substantial number of cognitively normal individuals at death will have significant levels of Alzheimer's disease pathology. So we've described these individuals as asymptomatic Alzheimer's disease. David Bennett and colleagues have described them in the ROS-MAP studies and called them high pathology controls. And to me, this group represents a very fascinating opportunity because we often focus almost entirely on understanding the molecular or biological basis of risk to Alzheimer's disease in terms of vulnerability to pathology. We don't focus so much on the molecular basis of resilience to Alzheimer's disease pathology. So what is it that confers resilience against developing symptoms in individuals who have appreciable levels of Alzheimer's pathology in the brain? So in our studies, we've included this group of individuals as an intermediate group between your controls and AD in order to address these questions. Question number 5 is, do these same protein and metabolite levels that change in the brain and are related to Alzheimer's disease pathology, do they also change in blood, and are they also associated with risk of developing Alzheimer's disease? And again, in these studies, we've used the same mass spectrometry-based methods that we've used in the brain to perform assays of protein and metabolite levels. Question number 6, once you identify a panel of proteins or metabolites in brain that you can also identify in blood that are also associated with risk for Alzheimer's disease, then you think this might be a plausible biological pathway mediating Alzheimer's disease pathogenesis. And then you try and test this in the relevant preclinical animal model. So I'm just going to run through one example of how we can put this paradigm in action. So we've used this study design to understand the metabolic basis of Alzheimer's disease. We've had a long-standing interest in understanding metabolic risk factors for Alzheimer's disease. We've been interested in glucose intolerance and insulin resistance, as well as midlife adiposity, showing that they're differentially associated with risk for Alzheimer's disease and AD pathology. So in these studies, what we've asked is, how do systemic abnormalities in metabolism mediate AD pathogenesis? And do peripheral metabolic signals reflect those in the brain? And can we relate peripheral signatures of abnormal metabolism to AD pathology and progression? So in this integrated metabolomics approach, we've used quantitative and targeted metabolomics to about 180 metabolites. These are mainly lipid species. They also include amino acids and acyl carnitines, and we'll talk about this in just a second. So small biochemicals, small metabolites are the end result of all regulatory complexity in the cell, and they represent regulatory complexity related to transcription, translation, as well as post-translation modifications. And we think metabolic changes are the most proximal reporters of alterations in the body in response to a disease process. So in this study design, what we've done is we've followed each of those 6 questions that I've walked through. So we first undertake a brain metabolomic analysis in these 3 groups of individuals: Alzheimer's disease, control, and asymptomatic Alzheimer's disease. And in each of these 3 brain regions, the inferior temporal cortex, the middle frontal cortex, as well as the cerebellum, and then ask if we can identify a panel of metabolites that differentiates brain regions in these 3 groups of individuals. So once we acquire this data, we analyze it using machine learning algorithms. We've typically used 2 machine learning algorithms, a random forest regression and support vector machine. And once we identify a panel of brain metabolites that are important in differentiating these groups of individuals, we then ask if we can assay these same metabolites in blood in 2 different cohorts of individuals that represent preclinical Alzheimer's disease. The Alzheimer's disease neuroimaging study that's enriched for individuals with mild cognitive impairment, and the BLSA that is enriched for individuals who all enter the study cognitively normal, but then along the course of follow-up, some of them develop Alzheimer's disease. We then proceed to test associations of these metabolites in blood with a variety of imaging phenotypes, with a variety of CSF-based assays related to Alzheimer's disease pathology, as well as longitudinal changes in cognitive performance. So these are the demographic characteristics of our brain cohort. So we had 44 samples divided into control Alzheimer's disease and asymptomatic Alzheimer's disease. The groups are well balanced for age, sex, and the ApoE genotype. In our blood cohort, the BLSA study consists of about 200 individuals divided into converters and non-converters. So converters are cognitively normal individuals who develop disease. Non-converters are those who stay cognitively normal. In the ADNI cohort, we had more than 760 samples. So these are enriched for participants with mild cognitive impairment. And again, these groups are well balanced for age and sex. So in our first step, we applied machine learning algorithms to ask if we could identify a panel of metabolites that differentiate groups of individuals in their brain tissue samples. And using these algorithms, we can come up with an accuracy of about 80% for a panel of metabolites that differentiates Alzheimer's disease from controls in the inferior temporal cortex. So these are the 26 metabolites that emerge from all of the 180 metabolites that we can assay in the brain. And as I've said, these are mainly lipid species. So these are mainly glycerophospholipids and sphingolipids. We also have some biogenic amines, amino acids, and acyl carnitines. The next couple of slides are now going to show you a selected sample of these metabolites and describe their associations with some of the phenotypes that we are interested in. So in each of these columns, you see one particular metabolite. So here you have this particular sphingolipid. Here you have a glycerophospholipid, and here you have an acyl carnitine. The first row of box plots represents their associations in terms of group differences between control, Alzheimer's disease, and ASYMAT. And what I'd like you to appreciate here is that in each one of these analyses, the ASYMAT group falls in between controls and Alzheimer's disease, suggesting that these metabolite levels in the brain do in fact represent some kind of a transition from normal to pathological states. In the middle row here, you have associations between brain tissue concentrations of each of these metabolites and CRAD scores that represent the total amyloid burden. And in the bottom row, you have the associations with BRAC scores that represent the total neurofibrillary pathology. The next analysis, we can take these metabolites and assay them in serum. And here again, you have these same three metabolites that I've just mentioned. These are log transformed concentrations, so even though the box plots don't appear to be significant, they are. You can see significant differences in the concentrations of these metabolites in the serum, and they're also associated with differential risk for progression to incident Alzheimer's disease in individuals who are initially cognitively normal. We can then ask if the serum concentrations of these same metabolites are also associated with brain-related, with MRI-derived measures of brain atrophy. And here we can use what we've called the SPARE AD index, which is an individual score of how Alzheimer's-like a particular person's brain atrophy patterns are. A higher SPARE AD index indicates a more Alzheimer-like brain. A lower SPARE AD index indicates a more control-like brain. And again, for each of these metabolites, we see significant associations between serum concentrations and the extent of Alzheimer's-like brain atrophy patterns. We can then do the same analyses looking at serum concentrations of these metabolites with CSF measures of tau and A-beta. So CSF measures of A-beta 42, total tau, and phospho-tau are well accepted biomarkers representing the pathophysiological signature of Alzheimer's disease. And again, each one of these columns here represents an individual metabolite, and each of these rows represents here concentrations of CSF A-beta 42, concentrations of total tau, and concentrations of phospho-tau. Again, showing that you can actually measure serum concentrations of distinct metabolites and relate them to CSF measures of Alzheimer's-related pathology. After having done these analyses, we were also interested in summarizing these associations. So we went from 180 metabolites, did machine learning, came up with a panel of 26 metabolites, and each one of these metabolites seems to be associated with a handful of endophenotypes. So how do we summarize these associations? So we've come up with what we've called the endophenotype association score in early Alzheimer's disease, or EASE-80, to summarize these associations. So what we've done here is generated a heat map. The heat map is divided into 3 sections. The first section represents the association of each of these individual metabolites here with brain-related endophenotypes, so group differences, associations with CRAD scores, and associations with BRAC scores. The middle section here represents associations between serum concentrations of these metabolites and endophenotypes, such as the imaging measures I've shown you in the ADNI dataset. And the last 2 columns here represents associations of these metabolites with phenotypes from the PLSA. Individual cells in red indicate significant associations such that higher concentrations are associated with a more Alzheimer's disease-like phenotype. In green indicate the other direction of association. We've described this as the EASE-80 score. Making abbreviations is usually very fun. We went through a couple of iterations. So this was one of the early contenders, blood and brain endophenotype score for Alzheimer's disease, but we decided that it was not going to be very politically correct. So we stuck with EASE-80. Finally, what we can do once you summarize associations of these metabolites with distinct AD-related phenotypes is to then map them onto known biological pathways that we know are important in disease mediation. So here what we've done is we've used distinct metabolite classes. So as I've said, we have glycerophospholipids, lysophospholipids that are membrane-bound. We have sphingolipids. So glycerophospholipids and lysophosphatidylcholines are membrane-bound metabolites. They can alter intracellular calcium levels, mediate tau phosphorylation through tau kinases. Sphingolipids and ceramides, on the other hand, are integral components of lipid rafts. They can modulate the microenvironment of enzymes related to APP processing. They can also signal through neuronal apoptosis mechanisms through a variety of second messengers. So each one of these metabolite classes then begins to impact very early upstream events related to the ultimate generation of the pathological features of Alzheimer's disease, including plaques, tangles, and brain atrophy. So in summary, what we've done here is proposed a novel paradigm and what we think is a novel study design that uses quantitative and targeted metabolomic analyses of both brain and blood tissue samples. So we've used more than 900 serum samples to identify distinct metabolite signatures associated with Alzheimer's disease. We've identified distinct metabolites belonging to the sphingolipid, glycerophospholipid, and acylcarnitine classes that we think are related to severity of AD pathology and progression during the preclinical stages of disease. And finally, we've proposed the EASE-80 score to represent the cumulative associations of each metabolite with outcome measures that are related to AD pathology and progression in 2 independent cohorts of individuals. I'm going to stop there. I'm going to acknowledge numerous colleagues and collaborators who've made this work possible. I'm especially grateful to participants in the BLSA for their invaluable contributions. And I almost always end with this shameless plug. If you or anyone you know in your network know of graduating students looking for postdoctoral fellowships, please ask them to be in touch with us. Thank you very much.